Hello, everyone, and welcome to our session on the intestine-chip. I'm Danny Levner, Emulate's Chief Technology Officer and co-founder. In this session, we want to highlight our evolving strategy around the intestine, particularly our expanding focus from small molecules to biologics drug discovery. And then we'll highlight a few specific applications of this technology. In this session, we have 3 speakers lined up for you. We're going to keep the question and answer period for the end of the presentation, which means that if you can put your questions using this platform on the right side of the screen as you did in the previous session, and we'll pick them up at the end. Our first speaker is going to be Luke Demasi, who is the Senior Director of Product Management here at Emulate. Luke is responsible for aligning a product innovation and strategy to the market and customer needs. Luke joined Emulate last year after 15 years in product development leadership. Prior to joining Emulate, Luke held positions at Agilent Technologies in the Cell Analysis Division, as well as in Corning Life Sciences, managing a portfolio of advanced in vitro products. He also has extensive experience commercializing and managing product lines supporting biologics drug development and manufacturing. In his presentation, Luke is going to discuss new strategy relating to biologics development and to highlight the product roadmap relating to the intestine-chip specifically. Luke?
Thank you, Danny. Let me just share my screen. Okay, can you guys see this? Still looking good? Okay. Is that good, Danny? All right. Thank you, guys. Perfect. Thanks a lot. Welcome to the breakout session today. My name is, as Danny mentioned, is Luke Demasi. I head up the product management function here at Emulate. In today's session, we're going to be highlighting one of the strategic areas of focus for Emulate, and that is our intestine organ models. We'll be showcasing some application data later on in the session pertaining to one of our more exciting models that we're launching over the next few weeks, and that is our colon intestine-chip. And my colleagues Narcia and Jordan will be taking us through a couple of case studies and user case applications of that particular organ chip model. Before we dive into that, though, I thought what I would do is I'll give a brief introduction to Emulate in case anyone here doesn't know what Emulate does or what the product line entails. As Danny mentioned, we'll also go through some of our strategic roadmap in terms of new models and applications that we're looking to launch over the next months and years. Okay, but before that, let's talk about one of the biggest problems that we see out there in the industry, and it's pertaining to the drug discovery process. We believe, and it's not just us, but it is well understood that the drug discovery and development process is fundamentally broken. It takes about 14 years to get a drug to market from discovery at a cost of about $2 billion. Not only that, the problem here is that 90% of these drug candidates fail in the clinical trials. And this is a very expensive part of the process to fail. So you can see that not only is it unsustainable from an investment perspective from pharma and biopharma, but also it ends up driving healthcare costs up for everybody. And we believe it can be fixed. And this is a unique opportunity for not only Emulate, but for organ-on-a-chip technology in general to really add some value in the preclinical phase before getting to clinical trials, which we are calling the in vitro to in vivo divide. So what's happening right now in that space? So in terms of the preclinical toolbox that our pharma and biopharma partners use today, they use in vitro cell culture methods as well as small animal and even non-human primate models to validate targets and to also test drug molecules for both efficacy and safety. Although cell culture methods have been very successful in terms of identifying new candidates, they can be used in screening modalities to test those candidates. They really lack some of that dynamic environment and the dynamic cellular microenvironment, which enables people to go into the mechanistic analysis study of how either the biology works or how these drugs work as well. Also, there's an understood translational gap between these in vitro cell culture models and what happens in the body. For animals, same thing. It's well documented that there's a translation gap there. Sometimes the biology in small rodents is just different. sometimes the pathways and the markers that we're trying to go after just aren't expressed. So here at Emulate, we believe that in a future of delivering true and integrated human biology to the bench, really taking in vitro cell culture to the next level, where we can start to recapitulate the complexity of the cellular microenvironment. This, in turn, will help us minimize or replace animal studies and also accelerate clinical development. So for those, again, who aren't familiar with Emulate, our human emulation system is delivered through a number of different product types and classes. So first, we have our consumable organ chips themselves. That's where the biology occurs. We have instrumentation that controls that biology. So these instruments are integrated and they provide an easy to use interface to provide flow, stretch, and power to drive that biology. And we also have software tools that augment the workflow with standardized protocols, protocol management, as well as data analysis tools there to help the workflow. At the core of Emulate's technology are our organ chips. And this is what is our first chip on market, which was the S1 chip. As you can see, it is transparent, it is flexible, and it's really where all the biology and all the action occurs. So I'll try this video to see if it will work for us to go in a little bit deeper. Here you can see from this animation that at the core of the chip, there are two channels. The top channel, which houses the epithelial side of an organ model, and the bottom channel for the endothelial side of the organ model. Both these channels are separated by a flexible but porous membrane. 7-micron pores enable channel-to-channel crosstalk, but also the infiltration of immune cells as well, or other types of cells. You can seed your chips in either one of those channels, and then we can apply vacuum to give stretch to the entire model. And this recapitulates things like peristalsis that you exhibit, that's exhibited in the gut, or breathing in the lungs. So it's that mechanical force. In addition to this mechanical force, the chips also enable the flow of independent fluids that are relevant for each of those cells that are growing, and it also enables downstream analysis of the effluent that comes off of the model there, so you can get all that data that comes off. To recap, our organ chip models provide that tissue-to-tissue interface, the epithelial-endothelial interface across that porous membrane that can be ECM coded. Our system enables a dynamic environment with independent fluid flow to each of those channels, as well as mechanical forces. And our customers can add complexity to the models. Not only can you add endothelium and epithelium, but you can also add circulating immune cells, stroma cells, microbes, hypoxia. And our system really is enabled to enable all that flexibility. One of our first organ models that we brought to market was our liver chip. And this comes in 3 flavors, either co, tri, or quad culture. This graphic here represents the quad culture liver chip that we have right now. In the epithelial, the top channel, you can see we have our human hepatocytes. On the endothelial, the bottom channel side, our human endothelial cells. And then we've also complexed in stellate cells and immune cells, the Cooper cells here, which are relevant for liver biology. We've validated this model over a number of years with our pharma and academic partners. As I mentioned, we have 3 flavors of the liver chip. Not only do we provide our customers with a standardized liver chip model, but we've also demonstrated that rat liver chips and dog liver chips can also be built on the system. When you build this complex biology, you're then able to understand or recreate the responses that people have seen in clinic. And here is an example that was published in 2019 with a compound called fieluridine. This compound was showing promise as a hepatitis B treatment. In preclinical animal studies, there was no evidence of toxicity or adverse effects. However, when they got to clinic, they saw catastrophic toxicity caused by the molecule, where 5 out of 15 of the patients died from liver failure. And again, this toxicity was not exhibited or picked up in earlier preclinical animal studies. In the paper here that I have, again, we can share this with you afterwards, we demonstrate how the organ chip system is able to pick up that species-specific in vivo response. So we're able to see toxicity markers at certain concentrations of fluoridine in the human chip model, and we didn't see those types of toxicities in the rat liver chip model. So again, when you build up that complex biology, we're able to demonstrate that the organ chip models are able to recapitulate and recreate those in vivo relevant responses. So that's just an example of one of our older organ chip models. Let's talk about what we're building now. And our strategy here at Emulate is really to deliver standardized, validated organ models and applications to our research and biopharma community. Over the past few years, we've had a tremendous experience to work with some of our academic, government, and pharma leaders. Our system is pretty much open-ended, so you can bring or build any biology on it. This is what we're calling the enabled models. And we've worked, as I mentioned, with our partners here to build a range of these models over the years, everything from ileum to colon to lung. What we do here at Emulate, we of course help our customers achieve the biology they want with their particular cells. But where we see a broad appeal of a particular model and an unmet need, we want to bring it in-house and standardize and validate that model so we can scale it out to our research and pharma community. And this is what's led to what we call our supported models and applications. So in 2018, we launched our first supported model, which is the liver chip. And again, there's 3 flavors, as I mentioned previously, really target at the ADME-tox space. In 2019, we added intestine chip, the duodenum chip, as well as our kidney chip, again, targeting ADME-tox space. And last year, we enabled models for lung chip. This includes for ADME and toxicity, but mainly we're seeing a pull there for infectious disease, specifically SARS-CoV-2 research. This year, we're really excited to be launching a couple of new organ models to our install base. And this includes our intestine chip, our colon intestine chip, as well as our brain chip, which recapitulates that neurovascular unit. Not only will we be launching these models, but we'll also be validating a number of applications around them. So these include immunology and inflammation, and that's what Nasia later on will be speaking to, immune cell recruitment, drug delivery, neuroinflammation, microbiome, as well as tumor models as well. The way that Emulate delivers these validated organ models is with our Emulate Bio kits. And here, as I mentioned, we validate the organ model, we validate those protocols, we qualify that cell material, and then we package it up and provide that for sale for our customers. Usually it comes in 12 chips here and have the corresponding pods that the chips go in, that go into the instrument, but we also include the biology, the cells themselves. So available now, we have our liver, kidney, and duodenum chips and chip material. This year, we'll be launching our colon, brain neurovascular unit, and lung small airway as part of the Emulate Bio kit offering. So that's a bit of background on the Emulate product line. Now I want to just pivot now to the colon intestine chip, which is our new model that we're going to be launching in the upcoming weeks. And Nasia and Jordan are going to be taking us through, as I mentioned, a couple of interesting use cases and applications where we are leveraging this chip. And I don't want to take away their thunder here, but the chip at its core is built based on primary organoids, gut organoids, derived from the organ. Now, we see organoids as an excellent source of the necessary gut cell types that are present. They're able to be expanded sustainably, and we're able to produce these consistently to be able to deliver a consistent and reproducible model to our customers. Just quickly on the workflow, we take these intact organoids, we fragment them, and then we seed them on that top channel. After we seed them and we add endothelial cells to it, we're able to see these organoids mature to become more in vivo-like, not just functionally and visually, but also gene expression patterns as well. This method was built on one of our earlier intestine chips, the duodenum intestine chip. And here you can see from this scanning electron microgram, the structure that we're able to recapitulate with our organ models. So the endothelium, with the endothelium present, we're able to see the proper polarization of those epithelium. Flow improves microvilli and villi formation, and stretch improves goblet cell production. And as I mentioned, not only do we see these structural, visual, and functional benefits of what the chip confers, but also in gene expression starts to map closer to in vivo-like tissue. So with the ability to build better models, where can we aim this technology at? And as I mentioned, Nasia is going to be taking us through one of the first applications, and that is immune-mediated barrier disruption of the colon model. And one of the disease areas where this is important is in inflammatory bowel disease, where you get that leaky gut occurring. IBD affects 1.6 million patients in the United States alone, 70,000 new cases a year. Interestingly, 55% of these cases fail to respond to therapy. And those that do respond, 80% fail to enter full remission. So it's clear that there's a problem here. And with better models, we can enable more effective targeting, enabling our academic and biopharma partners to discover and validate novel inflammatory pathways for the development of targeted therapeutics. In the second talk that Jordan's going to be giving, he's going to be discussing how we leveraged our colon organ chip to develop a human-relevant preclinical model to predict safety and efficacy of T-cell bispecific antibody. TCBs, as well as other T-cell engaging biologics, are tremendously potent, powerful. If misdirected, they can cause significant off-target effect. In terms of the discovery and development value chain, it's that preclinical development phase where researchers start to assess the specificity of these biologics, as well as the safety profile of these biologics. They use in silico and in vitro methods to inform downstream in vivo and animal testing modalities, including small animal as well as non-human primates. The issue here is that at times some of the targets just are not expressed in these model systems, leaving a gap for customers and researchers and developers to really figure out a way to assess on-target off-tumor effect. Finally, I'll leave you guys with just one other thought here in terms of adding additional complexity to this model. And that is, and I mentioned in previous slides, that you can layer in the immune cell component and look at how immune cells are able to traverse the endothelial-epithelial interface there. And this is important for both of these application areas. For inflammation, not only do we want to see what the effect of cytokines have on the epithelium and the endothelium, but the recruitment of immune cells like T cells from the bloodstream into the luminal space are also implicated in exacerbating the disease. A mechanism to understand, to quantify and analyze that effect gives our customers more targets to go after and really to start to look at the mechanism of this biology. Similarly, with biologic safety, you can see there that this particular modality here, you were able to look at applying the biologic to the cells on the endothelial side and looking at the effect on the epithelium. And with that, I will leave you guys and introduce Nasia to take us forward with our first application around colon. Thank you.
All right, thank you, Luke. Our next speaker, as Luke mentioned, is Nasia Apostolou, who has been part of the Emily team for several years now as a research scholar. Nasia led the efforts around developing the biopsy-derived colon intestine-chip that she's going to tell you about, as well as involved in running one of the partnerships around this project. Specifically, Nasia has been studying mechanisms that drive leaky gut syndrome in the human version of the chip. Prior to joining Emulate, Nasia studied the effects of stressors on the intestine and the intestinal homeostasis in animal models. Nasia received her PhD in experimental physiology for the medical school at the University of Athens, and she's authored a number of papers and patents. And then without further ado, Nasia is going to tell you about this biopsy-derived colon intestine-chip. Nasia?
So, good evening to all, and thank you for taking the time to attend this presentation today. This evening, I'm going to be talking about the establishment of a biopsy-derived colon intestine-chip and the application of it on studying leaky gut in humans. In this study, here we combined human biopsy-derived colonoids and tissue-specific microvascular endothelial cells to establish a primary derived colon intestine-chip.
Nancy, sorry to interrupt you, but your slides are not yet visible.
Could you repeat, Danny?
Your slides are not yet visible.
I can see them.
I can see them, Danny.
Oh, I apologize. Sorry.
So the epithelial cells reside in the top channel of the chip, whereas the bottom channel is lined with tissue-specific microvascular endothelial cells. Lateral to those, there are two vacuum channels where stretch is applied when and if needed. The chip provides the unique advantage of studying cell-cell interaction by co-culturing epithelial and endothelial cells in a microenvironment that has mechanical forces and stimuli similar to those that the cells sense in the body. This unique combination allows to the biopsy-derived epithelial cells to establish a monolayer with cytoarchitecture and transcriptomic signature similar to that one of the tissue. The on-chip culture, and specifically the presence of the vascular component, enhanced the establishment of a mature brass border and of a tight epithelial barrier as it was assessed by the apparent permeability to 3 kilodalton dextran, shown on the right-hand side of the slide. Understanding the complexity of the platform, we aimed to investigate how each one of the culture components affect the gene expression and signaling pathways at various time points of the culture. Hence, we moved to a more high throughput analysis at the transcriptome level, and indeed, RNA sequencing revealed that endothelium is the key driving force that differentiates the transcriptomic signature of epithelial cells as compared to colonoids in suspension culture. And that was also reflected on the upregulation of pathways that were related to epithelial cell differentiation as well as functional maturation. The colon intestine-chip consists of all the known basic epithelial cell types of both the absorptive and the secretory lineage, and the addition of the vascular component enhances the differentiation of the biopsy-derived epithelial cells, and at the same time reduces the stemness capacity, as it is expected based on the lifespan of intestinal epithelium. The homeostasis now of intestinal mucosa is shaped by a complex cascade of cytokines secreted by both epithelial cells and the rest of the surrounding stromal and immune components. To assess how responsive the colon intestine-chip is to inflammatory cues and how well it captures the signaling cascade, we used interferon gamma, a prototype and well-characterized cytokine on disrupting intestinal epithelial barrier, and IL-22, a cytokine the role of which in epithelial inflammation and restitution is still ambiguous. As shown by the FPKM values, the on-chip culture enhanced the expression of both pro-inflammatory cytokines receptors as compared to organoids in suspension. And now let's move on how the interferon gamma mechanism of action was modeled in the chip. Interferon gamma disrupts the intestinal epithelial barrier via two distinct mechanisms. The first one is the induction of apoptosis with parallel inhibition of epithelial proliferation, and the second one is the degeneration of the apical tight junctional complex structure. When stimulated basolaterally with interferon gamma, the colon intestine-chip lost the epithelial barrier integrity in a concentration, time, and donor-dependent manner. The tight junctions protein Claudin-4, occludin, and ecadherin were gradually internalized as the concentration of the cytokine increased, and the proteins zonula occludin-1 and the cytoskeletal protein effactin were relocalized as described in previous studies and models. The collapse of the epithelial barrier integrity was also coupled with decreased epithelial confluency 48 hours post-stimulation, coupled with activation of apoptosis, as it was confirmed by the increased cleavage of caspase-3 across all the three donors tested. In parallel, we assessed the secretion of pro-inflammatory cytokines and chemokines in the effluent media of both channels. Here you can see as a heat map representation the statistically significant changes in the secretion levels of these molecules. What this slide tells us is that, first of all, the colon intestine-chip is able to capture the secretion of all the pro-inflammatory molecules that characterize the acute inflammatory response. Secondly, it shows us that it can capture the polarized secretion of these molecules, as it is shown by the basolateral secretion of IL-6 and VCAM1, and the apical secretion of ICAM1. And lastly, and most importantly, with the example of the CRP, it can capture the donor-to-donor variability, which is observed in clinic. Having confirmed the reproducibility of the interferon gamma-driven barrier disruption, we aimed to leverage the same principles to investigate the role of IL-22 in this mechanism. IL-22 is a cytokine implicated in the progression of IBD. The data that have been collected so far in animal models advocate for an anti-inflammatory role of the cytokine through STAT3-driven induction of epithelial proliferation and secretion of antimicrobial peptides. However, more recent studies in human models, such as CACO2 and human colonoids, indicate the upregulation of the porous tight junction protein Claudin-2, resulting on a leaky barrier, and the induction of ER and oxidative stress, which results in cell death via apoptosis and pyroptosis. The colon intestine-chip captured the IL-22 signaling events. STAT3, which is the main downstream target of the cytokine, was activated on a concentration-dependent manner. We observed the highest activation 1 hour post-stimulation, and STAT3 remained activated at lower levels over the course of the culture. At the same time, interferon gamma failed to activate STAT3, a fact that highlighted the specificity of the response. The chip responded counterintuitive to the existing animal model studies. Administration of IL-22 disrupted the epithelial barrier integrity and morphology and induced apoptosis reminiscent of the response of the chip to interferon gamma. At this point, we aim to add an additional level of complexity by administering IL-22 binding protein. IL-22BP is the soluble receptor of IL-22 with a higher affinity for the cytokine as compared to the transmembrane receptor. In vivo, it is secreted by a subset of dendritic cells and timely regulates the extent of action of IL-22 on intestinal epithelium. The presence of the IL-22BP abrogated both effects, highlighting again the specificity of the response. RNA sequencing analysis and subsequent differential gene expression analysis verified the increased expression of genes related to ER stress and apoptosis, which was also reflected on the respective signaling pathways. Finally, aiming to establish a protocol where we would be able to study the recovery of the epithelial barrier upon the interferon gamma-driven injury, we minimized the exposure to interferon gamma to 36 hours and significantly limited the activation of apoptosis and extensive loss of the epithelial monolayer confluency, and employed tofacitinib, an FDA-approved JAK inhibitor, which is already used as a late-stage therapeutic in IBD. Continuous treatment of the chip with tofacitinib abrogated the interferon gamma-mediated increase of the epithelial apparent permeability, the relocalization of zonulocludin-1 and defactive that indicates the generation of the tight junctions, and significantly decreased the secretion of ICAM1, VCAM1, and IL-6 molecules that are associated with the acute inflammatory response. So to sum up, the colon intestine-chip is a platform that provides the unique advantage to study cell-cell interaction in a microenvironment with mechanical forces relevant to those one addressed in the tissue. This allows the biopsy-derived epithelial cells to develop into a monolayer with cytoarchitecture and transcriptome profile of reminiscent of that one of the tissue. The chip captures the mechanism of action of interferon gamma as it has been previously described and answers to tofacitinib, an FDA-approved JAK inhibitor, which is employed as a late-stage therapeutic in IBD. And lastly, the colon intestine-chip captures the signaling of IL-22, revealing an overall role of the cytokine and suggesting a rather detrimental than beneficial mechanism of action. Thank you very much for your attention.
All right, thank you, Nasya. Get my camera going. Oh, my camera is not turning on. Never mind. All right, thank you, Nasya. Our next speaker is Jordan Kearns, who is the director of the Applied Biosciences team at Emulate. Jordan, as part of this Applied Biosciences team, is responsible for the design and execution of studies that run the organ chip models for various applications across pharma drug development pipeline. Jordan also pioneered the use of the intestine chip in many aspects, including the incorporation of immune cells and developing its application to disease modeling and investigative toxicology. Prior to joining Emulate, Jordan worked with Professor Donning the Weiss Institute at Harvard, and through this work, he was awarded multiple awards, including the Grand Challenge Exploration Award from the Bill and Melinda Gates Foundation. Jordan received his PhD in biophysics and structural biology from Brandeis University, and he's been the author of multiple papers and patents. Jordan is going to speak about an exciting new application for the intestine chip, which is around the assessment of the safety of immuno-oncology reagents. Jordan?
Great. Can I just confirm that you can see my slides changing?
We can.
Okay, great. Thank you. So good afternoon, and thank you for joining me for the breakout session discussing organ chips for toxicology, investigative safety. As Danny mentioned, I'm Jordan Kearns, the director of the Applied Biosciences team at Emulate and a founding member of our scientific team. As we've covered in some of our earlier sessions, the lack of accurate human predictive preclinical tools is one of the major driving forces behind the high attrition rates of promising new therapeutics. We have cells cultured in hard plastic dishes do have limited biological functionality and relevance to in vivo physiology, while differences between preclinical animal species and humans often preclude accurate extrapolations to the clinic. So today I'm going to focus on an example of how our organ chip technology, specifically the Colon chip, can be used for evaluating the safety liability of an engineered T-cell bispecific antibody. So our ability to make accurate predictions of human responses relies on our ability to engineer the microenvironment of our organ chips to recreate key aspects of the cellular milieu human cells experience in vivo. Here we'll focus our work to validate an application to better predict the safety liabilities of novel engineered T-cell bispecific antibodies, which require the development of an immunocompetent colon chip with inclusion of peripheral immune cells. I'll also share the motivation of our pharma partner, Roche, to co-develop organ chip applications to be integrated as a more predictive tool with their toxicology project teams to assess preclinical safety of novel immunotherapies and our overarching goal to reduce their dependence on animals and to leverage human-relevant models to potentially reverse translate clinical findings and uncover the mechanistic basis for clinical observations. But first, a bit of context on the goal of immunomodulatory antibodies. The immune system is a complex network of different cell types that is responsible for the recognition and elimination of foreign substances from the body, including cancers. A helpful way to group human cancers is into primary tumor profiles according to immune cell accessibility. In the inflamed tumor, as represented here, the immune system is already active and has penetrated the primary tumor. This is the class that's most responsive to immunomodulation and T-cell killing. But in the other two profiles shown here, the immune excluded and the immune desert profiles, a key question is how do we encourage T-cells to better recognize and infiltrate tumors? So T-cell bispecific antibodies are able to physically link T cells through the CD3 receptor to tumors expressing target epitopes, and are a potent class of engineered immunotherapies that are particularly useful for patients with difficult to treat cancers. But we should also ask ourselves, what is the ultimate goal of an engineered therapeutic? So many patients don't respond well or have no benefit at all to drugs. Patient B, represented here. While other patients, patient A, the dose may be effective, but also cause significant on-target off-tumor cytotoxicity, because even normal healthy tissues can express low levels of cancer-linked antigens. In the clinic, a single population dose is often the goal, which balances the benefit of the treatment against the potential for side effects. In practice, a patient non-responsive to a treatment will have their dose adjusted until an effect is achieved. However, getting accurate data to inform these dosing decisions is a challenge in the absence of human-relevant preclinical tools to enable predictions. And this is a key limitation of engineered therapeutics, that normal variation in target expression causes toxicity and adverse events. So to understand and overcome the response variability of patients to medicine, we need better tools that enable us to determine which patient should receive a particular treatment, the right patient, and which dose should be given to each patient with the potential to respond, the right dose. While these engineered immunotherapeutic antibodies are promising, they are also complex. As we've discussed, the key concern is on-target off-tumor toxicity, where healthy tissues may express the target antigen at low levels, leading to unintended adverse events. Shown here are the results of a phase 1 clinical trial that showed striking efficacy, but also showed significant safety liability as well. Given the importance of on-target off-tumor toxicity and the potential for adverse events, we've developed a new application using the intestine chip to help improve our clinical evaluation of promising lead candidates. Just to remind you, here's an illustration of the intestine chip architecture that we're going to use. And there are many different flavors of epithelial cells that can be seeded onto our intestine chips, including iPS-derived or cell lines. But in this talk, I'm going to again focus on our primary intestine-derived from human biopsies, colonic biopsies. So now that we're oriented to the primary intestine chip, talk a little bit about the T-cell killing application that we've been developing. In this approach, we model an acute exposure of T cells with the TCBs to target expressing epithelial cells. First key component of TCB-dependent T-cell killing is the presence and expression of the cancer antigen, shown here. In the presence of both the target and the immune cell receptor, the TCB physically links the immune cell to the target, causing activation and release of the cytotoxic payload, represented here by granzyme B. This eventually leads to epithelial cell killing, as measured by cytokine release and apoptosis. Importantly, the application described here does not include the key components of immune cell circulation, recruitment, and diapedesis, as Luke was mentioning earlier in the talk. These elements are a part of a further development that will be released as a validated application later this year. So here we see a cross-sectional slice of our colon intestine-chip showing target characterization and expression and subcellular localization relative to a control sample. This confirms target expression, which is essential for TCP recognition and the subsequent cascade of immune responses. Well, we can certainly appreciate a beautiful image. You see that the subcellular polarization of the target can also be quantified using confocal microscopy. This is a Z-plane intensity analysis showing the correct apical polarization of the target and surface exposure, which is critical for TCB function, as it's in order to link the migrating immune cells to the target. So these results correlate with the in vivo expression of this particular target antigen. Interesting histopathological analysis of the target indicates that the expression in the healthy and gastrointestinal tract shows clear regional differences between small intestinal duodenum and the colon. Given the regional expression differences of the target, the one question that we have is our system sufficiently sensitive to capture the TCB-mediated toxicity in these different tissue types? So to test this, we also were able to compare the differential target expression and target-dependent responses in our colon and duodenum chips. You can see that the regional differences in target expression at the RNA level here that show that we see the regional differences that are not actually present in the three-dimensional organoids, but are recapitulated in the context of flow and stretch on the chip. The chip microenvironment is essential to be able to more accurately model the human physiology, as we can see in comparison to the in vivo RNA levels on the left. So how do we quantify the different readouts on the chip? Here we see a representative tiled image of a primary intestine chip showing 3 different fluorescent markers. We have our nuclei stained here in blue. We have apoptosis stained here with measured here with caspase 3-7 activity, and we have the PBMCs themselves labeled that we can count and see throughout the chip. And so using these images, we're able to confirm both the number of apoptotic cells and also to confirm T cell killing. But just one readout is not sufficient to determine the function of TCBs. We've established here previously that the target is expressed and polarized in the chips. We've shown that we can measure the epithelial responses from full tiled images of the chip. However, we also need to understand how the TCBs are directly affecting the immune cells themselves. So here we're measuring a dose-dependent increase in the early activation T cell marker CD69 from immune cells that were harvested directly from the chip after treatment. What we can see is relative to the target IgG and non-controlled TCBs, a TCB that's blind to the cancer antigen, is that the immune cells are specifically activated at concentrations that are relevant to the clinic. We're also able to show that 2 TCBs with different affinities to the antigen show differential responses. We have the binder 2 and the binder 1 represented here. This again helps to support the hypothesis that in these intestine chips with patient-specific epithelial target expression could be used to tune the therapeutic dose and TCB formulations, moving closer to patient-specific treatments. Here's the summary of the most relevant readouts we've determined to be predictive of TCB-mediated toxicity. In addition to the target expression and polarization and T cell activation that we've just shown, we've also performed a profile of pro-inflammatory cytokines, including T cell specific cytokines like interferon gamma and granzyme B. Taken together, these data, which were measured in multiple repeats in different patient donor samples, align with the signatures of cytotoxicity and adverse events that were observed in the clinic, indicating the utility and predictive potential of the application. Having validated some of these readouts, we can now return to the hypothesis that some of the adverse events that have been seen in the clinic could be related to regional differences in target expression between the small and the large intestine. The data here, I'm showing a comparison of the duodenum intestine chip here in the dotted lines and the colon chip represented here in the solid lines treated with TCBs, and shows a discriminating signal for immune activation for binder 1. It also shows that the binder 2 formulation, which was a higher affinity compound, is showing activation in both the tissue types, again indicating the potential to use organ chips to tune the safety profile of TCBs. Could it be that some of the signals that we're seeing here were missed in preclinical screening because the tools that were used didn't allow for the full maturation and differentiation of the epithelium? The correlation between the adverse events that were seen in the clinic and the results from our intestine chip here strongly suggests that our primary intestine chip models can be used to reverse translate and actually de-risk engineered therapeutics that are in the clinic. So that's all I have for today. I hope you enjoyed the session and I hope you enjoyed my talk. And thank you very much. I'll pass back to you.
All right, thank you very much, Jordan. At this point, we have about 7 minutes left for the question and answer period. I'm going to read off the questions and direct it to our panelist speakers. Let's start with the first question here, which is, were you able to mimic the microbiome complexity for your study to assess its effect on the epithelial barrier? Jordan, I think this one is up your alley.
Can't hear you, Jordan.
Sorry about that. So I think Don Ingber actually gave a very nice presentation earlier today on some of the work that they've done at the VIS. And we do have that on that full validated microbiome application on our roadmap. I think, Luke, I can pass it over to you in terms of how, when and how. But suffice to say, we do have some powerful capabilities in terms of modeling different bacterial compositions and oxygen control and other aspects.
Yeah, you said it, Jordan. I think people have been able to do this. The VC have been able to do this. We've done certain studies as well internally as a service, but we're looking to really standardize a protocol for this. Hopefully before the end of the year, but probably going to be in FY22.
All right, thank you. And I'll just add myself that in Dawn's studies, we were able to see the effect of the microbiome on the epithelial barrier integrity, both in terms of probiotics helping the barrier integrity, as well as pathogenic organisms harming the barrier integrity. Just to stay on the microbiome theme, I'll skip to a different question. In organs and chips with both cells and bacteria, how do you manage the needs of both in terms of oxygenation, specifically since the PDMS is permeable, etc.? Jordan, I think this one good for you.
Yeah. Okay. So I think we have a couple different levers there in terms of how to manage both. I think because it's permeable, we're able to obviously we get perfusion coming from the vascular side, and we're also able to have the environmental control over the whole system. Flow is obviously a key component of that as well. Hopefully that will, those are some of the levers and the tools that we have available to us to control this kind of factors.
All right. Let me direct the next question to Nastya. It's about ECM. I'm actually going to combine two different questions off the chat. There's a question here about what would an alternative to Matrigel be, or is there a better alternative to go to the cells? Separately, we have a question around ECM, how we choose ECM in our chips.
So our chips are coated with human-derived collagen 4 and fibronectin. Matrigel exists in a very low proportion in the coating of the top channel. Another approach that we would be definitely interested to incorporate, and it can be used, it is also, as the technology advances, the decellularized extracellular matrix that derives from biopsies.
All right, and in fact, it is a big part in our development of individual models to choose the correct ECM for either the healthy or diseased models. Okay, moving on to a different question. There's a question, I'm also going to combine a couple. There's a question that talks about whether we worked on different portions of the intestine, and let me combine that with the question around comparing the organs and chips with organoids. So what can we say about the difference or the advantages of cells within organ chips relative to just organoids? Nastya, maybe you can take this one.
So far, we have worked with 3 different regions of the gastrointestinal tract, and these are the duodenum, ileum, and the colon. It is possible, yes, to use the chip for patient stratification, since we have confirmed that it captures the donor-to-donor variability in terms of differentiation capacity under baseline conditions, as well as upon stimulation with pro-inflammatory cues. And the changes that we see compared to organoids alone are, as we mentioned, mainly driven by the presence of the vascular component. We do see significant differences in the maturation and differentiation of the absorptive enterocytes, but we haven't identified yet which is the paracrine or circulating factor that drives these specific changes.
Okay, I'll just add, we've done an extensive comparison of the organ chips to organoids in their regular form, and when we see advantages across absorption and inflammatory response and so on, we're happy to share papers and posters that we have on that. Okay, moving on to the next question. I think Jordan would like to take this one. Is your system able to recapitulate the lamina propria in any way? Has anybody been able to model Peyer's patches?
Well, the first part of the question, we do have some work in that regard. We are capable of embedding immune cells of different formulations. It's a complex question. It's a complex problem. So I'd say that we're still early in our abilities there, but yeah, we've been able to, and it's also really dependent on access to patient materials. So in the case of the resident immune cells, getting access to lamina propria samples, enough material to be seeding chips is can be a challenge. But we do have some quite nice results, and we do have some posters that we can share on that work as well. In terms of Peyer's patches, I don't know that we have anything specifically there, but perhaps Nasi, if you know, or maybe there's some material out of the V's, but not to my knowledge.
No,
All right. Sorry, go on, Nasa.
We haven't investigated the and whether M cells are present in the chip or not.
All right. Well, this brings us to the end of our allotted time, so we'll conclude the question and answer period. Thank you, everybody, for attending.
Thanks so much.